Welcome to WinelineRadio and WinelineTV. This is your host, Robert Scott. Today we have a very special discussion for you. Joining me today is Vouillamoz, a noted biologist, grape geneticist, and DNA profiler, and my good friend and collaborator, Luis Torres, president of the Wine Business Academy and founder of Go-Wine. This should be very interesting. Good morning, gentlemen.
Good morning.
And good morning, Robert. Thank you for your generous offer and your time this morning on WinelineTV and WinelineRadio. I guess what we'd like to do before we begin is we're just going to thank Dr. Vouillamoz. I had the pleasure of watching you present at the Climate Leadership Conference in Oporto, and it was very inspirational. You covered a lot of material in a very short amount of time. So again, it's glad to speak with you again.
Thank you very much. Well, I had to leave some space to Al Gore for speaking.
That's right. He needed a little more time, right?
Yes.
But no, it was a very effective conference, and the information that was derived from that is incredible. For those of you listening, just to let you know about the format today, we're going to be speaking with Dr. Vouillamoz, kind of in a back and forth Q&A. So Robert will be asking some of these questions, and I will as well. And then we look forward to this exceptional content from Dr. Vouillamoz. So I think what we'd like to start with, for the audience, that's listening, if you could just tell us, in a couple of minutes, a little bit about your trajectory, your curriculum.
Well, with pleasure. No problem. My name is José Vouillamoz. I'm from Switzerland. I'm a biologist, grape geneticist, and I was trained at UC Davis in grape genetics and DNA profiling with the professor Carol Meredith. And in my career, I've had many women that were very important for me. The other one is obviously Jancis Robinson, with whom I collaborated on the Oxford Companion to Wine, on the World Atlas of Wine. And after a few years of collaboration, we started a huge task, which was the writing and publication of Wine Grapes, the ultimate reference on all the grape varieties that are cultivated in the world.
Wow, that's fantastic. And I know today we're going to go ahead and close with that information. I can't wait to get to that. But I wanted to kind of go through like a pyramid of questions in this regard. According to your business profile, you conduct DNA profiling for your customers. And for many of us, we have to kind of walk before we run on this subject because we're new to it or have varying levels of expertise. DNA profiling, we were wondering, how does that help winemakers and viticulturists be more successful in their businesses?
It can help them in many ways, many different ways. First of all, when a winemaker has an unidentified variety in the vineyard, it's always important to know what they are cultivating because it can affect the taste of the wine. This is the first step. The second step, when you have to replant a vineyard, if it's a brand new one, and for example, you would like to plant Syrah, it happened to us in California back then, many years ago. A producer wanted to plant 10 or 20 hectares of Syrah, and he got material to plant it, and he was suspicious, so he gave it to us, and we analyzed and said, it's not Syrah that you're planting, it's something else. So we saved him a lot of time and money by identifying very early in the process what he was going to plant and what was wrong. And also, if you have a very old vineyard with different varieties and you want to replant it in a similar fashion, you need to know exactly what you have in the vineyard. So we help also maintaining the diversity, the biodiversity in the vineyards.
Okay, that's fantastic. And so there are obviously a lot of situations where they think they're planting one variety and in fact they're planting something else.
I would not say a lot, but it can happen, especially in the new world. In the old world, in Europe, they are more trained in identifying visually the varieties, but we consider that in nurseries and also in grape collections, we have between 3 and 4% of mistakes, of mislabeling or human errors that can happen. So with the DNA profiling, for example, we can solve these mistakes.
Fantastic.
Let me jump in here and just ask a quick question. You use genetic DNA markers to identify these vines, as one would do in identifying the historical data of a person, correct?
Yes.
Can you kind of take us through that process a little bit? How do you collect from the vine itself in order to identify these markers?
You can collect almost any part of the vine that you want, but the most convenient for me is to collect the tiniest leaves, the tiny leaves that are very young, because the tiny leaves contain a lot of DNA. A larger leaf contains as much as the DNA as the tiny. So then you dry this leaf, and you go through extraction process to extract the DNA, and then we look at specific portions of DNA that will be unique to the variety, but that will be different from one to the other. It's just like for human beings, we all have a single unique DNA profile that is different from your brother, from your neighbor, from anyone else. But in all your body, it will be the same. So it works the same with the vines.
Well, it's very interesting to me because I recently, in fact, last Father's Day, I was given a gift of a DNA analysis for my history. And all these 70-some years, I have had this distinct knowledge through contact with other members of the family that my DNA was primarily Scottish, and yet it came back as 87% Northern European, which I guess supposes that the Vikings made their way into Scotland at some point. So I thought that was very, very interesting, and I'm sure that this kind of unique discovery on your part can be very telling to the producers of the wine grape.
Yes, of course. Well, for the ancestry analysis that has become a commercial purpose in the recent years, you get a general picture of where the DNA comes from. It's not so specific. If you want specific study of your family tree through DNA, it's not that easy because you need to have written agreement of every person that you will analyze the DNA of. So if you want to be more precise, you need to have a serious background for that. But yes, for the grape varieties, it's the same technique, and the discoveries can be fascinating when you understand that a vine in one place is the same as another variety in another place. You understand the migration of the people also, of the viticulture and the people. So it's genetics combined to history.
This new research published in Nature Plants on the findings of ancient DNA, I mean, that was significant. It was run all over the globe on the major publications. What do you think the findings, what were the most significant revelations in those findings, in your opinion?
Well, I find this new research fascinating. I'm almost jealous I was not part of it. But to tell the truth, I had the same idea more than 15 years ago, using the same technique, but it was not developed enough back then, and it was way too expensive. They were using what we call SNPs. It's single nucleotide polymorphism. So it's the variation at one single molecule in the DNA that you're looking at. And this is what they have used for these archaeological samples coming from France. And the result for me is really fascinating and amazing, because they were able to find one grape pip near Orléans in the central France that is identical with the modern grape called Savagnin Blanc that is used to make the vin jaune in the Jura. We knew that this grape was probably very, very old, but we always had doubts because if you read an ancient document of the Middle Age period and they say Savagnin or something, you cannot be 100% sure that it was exactly the same as the Savagnin we are still cultivating today. It could be the ancestors or the cousins or the brothers or sisters. Now we know that it was exactly the same. They were cultivating 900 years ago at least. And this is a groundbreaking discovery.
Yeah, that was fascinating because obviously here in the States, we don't focus that much on the Vin Jaune and the Sauvignon Blanc. And a lot of people, of course, immediately confuse that with Sauvignon Blanc, right?
I know.
No, right? And so where do you see the future going with this discovery?
I wish that they will expand their research to other archaeological sites all over the ancient world. I myself come from Switzerland, where we have grape remains. We have grape pips coming from archaeological sites that are even older than what they had. Some of them date back to 800 years before Christ, BC. And it would be super fascinating to analyze the archaeological remains in the rest of Europe and also in what we call the cradle of viticulture in the Near East. Archaeological sites in eastern Turkey, in Armenia, in Georgia, also in Iran. These are places that I have already visited and I have studied back then together with Professor Patrick McGovern, who is an archaeologist that is specialized in analyzing the remains of wine traces in the jars that are found in the artifacts that are found in the archaeological sites. And now with this technique, we could discover fantastic results.
Great. Robert, do you want to pitch a question, or would you like me to continue?
Well, let me say this. With all your studies, you've been able to really pull down vine age to a very small margin of error. What causes that?
Well, this is a different topic.
Yes.
It's what we call Dendrochronology. So it works more or less the same as you would estimate the age of a tree. But for a tree, it's very easy. You can with the naked eyes, you can count the circles. It's also possible to do the same with an old grapevine, but it's too small to see it with your own eyes. You need to use a microscope, and you need to make super thin slices and use specific coloration in order to be able to count the circles. And I'm collaborating with a laboratory in Switzerland, and we were able to narrow down the age of a vine with less than 10% of mistake of margin error, which is very, very precise.
I would say. And does this really help the wine producer to be more successful in the vineyard? And as a follow-up to that, do you think older vines contribute to a better wine?
So we know many producers or even regions that are proud of the age of their old vines or old vineyards. I'm thinking of Chile, for example, where I've been several times, where they really have old vines, more than 100 or more than 150 years old, and they use it as a marketing tool in order to promote. Well, it is justified. I'm not criticizing, but it's good to use it. And also, we think generally that old vines give better wines because the roots go deeper in the soil, so you express the terroir much better. And the vines getting older will have naturally lower yields. So they usually make better wines. There are exceptions. Some recent vineyards can produce fantastic wines as well. But the general picture is that old vineyards are cherished by the producers and by the consumers as well. So it's very interesting for us to bring science to tell exactly how old the vines or the vineyards are.
That's absolutely fascinating.
So one follow-up on this now. So we move from the vine age. I'm going to shift back into what's going on in the world of genetic modification and genetic editing and the discovery of CRISPR technology, and specifically as it applies to grapes. So this is very intriguing, and we never take a position of support or non-support of GMO. We just like to hear how CRISPR technology will help grapes more resilient to disease, et cetera, potentially, if it is. And if you can just give us a quick note on an easy way to understand CRISPR technology.
Yes, first of all, it is not considered, or I would say not yet considered, as a GMO technique. And the international regulations still have to make a decision on that. It's not easy. It's not like what people know of the GMOs, where you can take, if you fancy it, if you can take a gene from a mouse and put it in an elephant, which would be quite possible. It's not the same. It's a natural process that bacteria have developed to fight against viruses, and we are simply mimicking what the bacteria have invented, and we apply it to other organisms, to human beings, of course, and to grapevines as well. And basically, it works like a pair of scissors. You can cut some portions of the DNA you don't like, or you can take one portion and stick it to another place, but you usually don't add anything, no foreign DNA into your organism. So it has a huge array of applications in human medicine, of course, and with the grapevine, it's currently under study. The research is very, very recent. The technique dates back to 2012, and the first results in grapevines were 2016, where they were able to modify some genes in Chardonnay, but only in plant cells cultures, not in actual plant. And only in 2018 they were able to have the tolerance against powdery mildew in vitro plant that can be developed into a growing plant. So it's really in the process, and this technique could help us to fight against the fungal diseases that are downy and powdery mildew, botrytis, to fight against drought, to fight against frost, because we now have the complete genome of the grapevine, and we understand where the genes are, the genes that are responsible for so-and-so characteristics. So, and now we can go there, and it's like Photoshop with the DNA. You can arrange a little bit what you don't like.
Well, is there any risk involved in applying, let's call it gene editing?
Well, to be honest, in grape genetics, I wouldn't say that there is a great risk of errors, but in human medicines, yes, of course, sure. Because this technique is not yet perfect. And sometimes when you want to modify a gene, a neighboring portion of the DNA can also be modified, and you didn't want that. So it can have serious effect in humans, and the technique is not yet used in routine. If you do it with grapevines, it's not a big deal, because if your vine is problematic, you just discard it and you do it on another vine. It's not that problematic. What I see as a future problem with grape genetics is that if you want to modify, let's say, Pinot or Syrah or whatever, and to have a Syrah that is resistant to fungal disease, you will do it only on one clone of Syrah or one clone of Pinot. And these varieties, especially Pinot, for example, that is very, very old, we know 1,000 different clones of Pinot in the old vineyards and in all the collections in the world. You should do that to almost all of the clones in order to maintain the biodiversity within the grape variety. And this is very important to me, because if you choose one clone saying, well, that's the best, and we will focus on this forever, it's very dangerous, because this clone can have future problems with the global warming. It can have future problems with new disease appearing. And you lose the diversity, you lose also the complexity of the wine that will be produced.
Well, that brings us really full circle to discussion of climate change and genetics. I was looking recently at a animated graphic that illustrated the rise in temperature over time as it relates to vineyards that will have to move north in order to maintain the type of concentrations and characteristics that are now producing at a lower level latitude. And it poses the question that in a number of years, who knows how often or how long, they'll be growing Cabernet Sauvignon in Sweden.
Well, it is true that with the global warming, we see the latitude and also the elevation of the vineyards moving upwards. You obviously know that in southern England, we are starting to produce very serious white wines, serious sparkling wines, and the big names in Champagne have already invested in southern England in order to produce excellent sparkling wines, which was not possible 30 or 50 years ago. So yes, we do have examples as well in Sweden, in Norway, of vineyards that are becoming more important. But in the traditional regions, I cannot imagine, at least not in my lifetime, that, for example, in Burgundy, they will abandon Pinot and plant Grenache or anything else. I cannot imagine that as a wine lover. What has been done and will be done in the future is to mitigate the effects by changing the viticulture technique, by changing the pruning, the irrigation, by changing the clones. And we have many viticultural techniques and means to try before we plant something else. And for me, one of the most important research that still needs to be done is to find what is the best rootstock for each clone of each variety. When people say we need to match the variety to the terroir, okay, it's nice, it's like poetry, but it's way too simple. It's way too simple. You need to match to each terroir. You need to match the right rootstock with the right clone, and you need to maintain the clonal diversity. And also another thing that I'm supporting is to go back to the ancient varieties that used to be cultivated in the place and that have been abandoned many times in favor of more famous varieties. Because if you take old local varieties that have been discarded recently, they've been through hundreds of years of different climates. So with this new change of climate, they are the best ones to cope with it.
That's really interesting. It seems to me that I've kind of seen that with a resurgence in the last decade of Tannat, for instance, which is a grape that kind of went out of favor in Bordeaux and other parts of France, and now has become a leading grape in Uruguay, for instance.
Yes,
that's a great example.
Yeah, but this dates back to the years before phylloxera, when the Tannat or Carmenère was introduced in South America, or Malbec, whose name is Isco. This is long before the global warming. It's not related to that. But that kind of grapes, for example, that were hard to ripen, especially Tannat or late ripening varieties. For example, in Switzerland, we have late ripening varieties. And up to now, the global warming has helped them to mature much better than they did before. So in some cases, global warming can have positive effects. In some cases, I'm saying.
Right. Well, I'd like to follow up with this then, because we know that your time is very, very valuable. There's really two questions to close, and thank you for this thorough explanation. I know a lot of the answers to the questions for our listeners here are going to be found in a very special place that I'll talk about in a second. So there's two questions here. Do you feel that there's some advances in genetics that'll make a higher quality wine possible? And then we really want to close up today with, introducing the listeners, if they haven't heard about it already, they should, to the book that you co-authored with Master of Wine Jancis Robinson and Julia Harding, entitled Wine Grapes. And I believe it's being translated into Japanese. Is that correct?
Yes, it's correct. It's due out on July 12th in Japanese. It's the first translation of the book, and it happens to be in Japanese. So I'm very much looking forward to holding the book in my hands. And I must remember to read it in the right sense, so from bottom to top.
Perfect. Well, so let's address those two things. I mean, advances in genetics that you feel could make higher quality wine, do you think? And then we can just touch on the subject matter of wine grapes.
Yes, of course, that genetics has always focused on making better wines. And now that we have the full genome of the grapevine, which happens to be Pinot, it was done in 2007. And for the history, in 2007, the cost of the research was 23 million euros for the full sequence of Pinot. If you do it now with the advance of the technique, it's $500. Wow. It's amazing. It's amazing. In 15 years, it has changed completely. And I find this fascinating. So the evolution of the research is really, really going fast. CRISPR-Cas9, this gene editing technique, is one of the future application. And now that we know most of the genes, where are they located in the genome and what are they coding for, we know most of it, or the major of them we know now. We can interfere with the genome in order to have varieties, to produce varieties that are more resistant to fungal disease. Or we can also use classical genetics with hybridization. You take the pollen of a vine, you put on the female part, the pistil of another vine, and you create new hybrids that are resistant to the fungal disease. And the general public is asking for a cleaner viticulture. They don't want the winemakers to spray a lot of nasty chemicals on the vineyards for the next 50 or 100 years. So that kind of advances in genetics will also help not only to have better wines, but also to have a cleaner viticulture.
Fantastic. That's great. And so this is very encouraging. Now, let's just look at the book. Tell us a quick cliff note of this. I believe it's a 1,400-page tome, and what you did in there.
I think it's 1,280 pages.
Okay,
it's close enough.
It's 3 kilos and 70 grams. It's like babies. We are very precise in the way of the baby. It took us 4 years to make that, and to go back in history, in 2008, I had contacted Jancis after working with her for a few years, and I suggested her to make a small book about the history and origin of the major grape varieties. I had in my head the 15 more important ones, and she is extremely clever, and she had a brilliant idea, saying, no, we need to make a book about all the grape varieties. And I did not take time to calculate what difference it would make. I had to say yes. Instantly,
Wow.
I said yes. I did not realize that it would take us 4 years to produce this huge reference book. But we're very happy we've done that because it had a significant impact in the wine world. And we hope that we have also helped maintaining or even resurrecting some old varieties that had been abandoned. And we also hope to be able to make a new edition in a few years because things are moving so fast. New information, new discoveries, new hybrids or crossings, ancient varieties that have been recuperated. A lot of things have already changed since the release in 2012, and we are much looking forward to releasing a new edition we don't have any number, maybe in 3, 4, 5 years, we don't know.
Well, that's fascinating. I can't wait to get an audio book on your tome. And the next time we speak, I would like to have a discussion about one of my favorite wine grapes, one that I am totally fascinated with, and that's Sagrantino.
Yeah, the history of Sagrantino is really amazing. It's a one-man rescue story. It's Arnaldo Caprai who was producing this variety near Montefalco in central Italy. And this variety is very powerful, has strong tannins, and the local people were not believing really in this variety. And he was the one to believe in it. And he was strong enough to promote it and also to promote it abroad. He went to the United States and he struggled also to have it in the appellation. And now we have an appellation, Sagrantino di Montefalco, based on the work and on the passion of one single man. And this is fantastic because for me it shows that the varieties that are today a little bit obscure or misconsidered, they might be the flagships of tomorrow. So you must never abandon a grape variety because with global warming, maybe in 50 years from now, it will be a star. We never know. So each grape variety has a right to live.
Well, Dr. Vouillamoz, thank you so much. You've really added a upper level to our usual discussions, and I found it very interesting and fascinating, and it really opens up many more questions to be asked. Wouldn't you think so, Luis?
Yes, I'd like to chime in and echo, first of all, our gratitude for you to be here. They can find you on the web, josevouillamoz.com. Is that correct?
Yes, it's correct. And also about the book, we have a lot of information on winegrapes.org.
Exactly. And then you are on Twitter as Jose Grapes?
Yes.
Okay, perfect.
That's correct.
If the listeners would like to find a lot more, please reach out to Dr. Vouillamoz. On behalf of Go Wine, we want to thank Robert for doing this today because, again, you have a busy day. And certainly not the least, Dr. Vouillamoz, we look forward to speaking with you and sharing this content across the world.
Thank you. Thank you very much. It was a real pleasure for me.
Well, and I must say that I look forward to the next conversation. This is your host, Robert Scott, saying many thanks to Dr. Jose Vouillamoz and to Luis Torres for this fantastic interview. I think we are just beginning to scratch the surface of DNA. So hopefully more to come in the future. Thank you very much, gentlemen.